Good morning. I would like to present our next speaker, Dr. Linda Chalker-Scott. She has spoken with Think Trees for over a dozen times. Dr. Chalker-Scott has a PhD in horticulture from Oregon State University and is Washington State University's Extension Urban Horticulturist and Emerita Professor in the Department of Horticulture. She holds ISA credentials as a certified arborist and track. Additionally, she is ASCA consulting arborist. Linda conducts research in applied plant and soil sciences, publishing the results in scientific articles and university extension fact sheets. She is also actively involved in enhancing the scientific literacy of her audiences by teaching them how to assess the credibility of information, which is so important now, from the print and online sources. Thank you so much, and we're so happy for you to be here.
Thank you, Elizabeth. Appreciate that. Well, thanks very much. As I've been telling lots of people, I am retired and I get to do the things I love, not the things I have to do, and I love being here at this conference. So thank you for having me again. Appreciate it. Okay, so there is a handout for this. Everybody have a handout? If you don't, there are extra ones floating around, and if you raise your hand, I think that maybe you can get one. If you don't get one because there aren't enough copies, I know that Marissa's going to share it in chat with you, so you can get a copy of it and download it. Okay, so this is one of my favorite talks. It's always changing, and the interesting thing about the diagnostic delusions is just not people who don't know the science, don't know the profession. It's all of us. And so one of the things I'm going to talk about is something that fooled myself and many scientists for a long time, in part. So that one you get to look forward to. But this is a lot of fun, and it will make you look at things differently and think twice before deciding what a problem is. So first of all, and this is on the sheet, it's kind of a little checklist of things you need to have before you make a diagnosis. So obviously you need to have a soil test. Maybe it's not obvious, but you should have a soil test. As one of the examples we're going to discuss today will show, if you don't know what's in your soil, you cannot diagnose a nutrient deficiency. You can't. I mean, it would be like going to a doctor and not having a blood test, and then the doctor would sit there and tell you all the things wrong with you when you have no idea what your blood test is showing. Species information. When you're looking at plants from where you're located, perhaps your native plant palete's pretty small. Perhaps your plant palette period is pretty small, but sometimes you can be someplace like where I'm from, where we've got plants not only from our own region, but lots of things from all over the world. And so you really do need to know your plant before you make a diagnosis. You need to understand the environment. Are there, pots of cold temperature? In other words, something that's down at the bottom of the hill is often going to be colder than at the top of the hill because cold air falls and ends up kind of getting collected at the bottom of the hill. You need to know how a tree was installed, and you need to know how it was managed over at least the last few years, if not longer. Lots of problems that show up in the trees have been caused by damage that was done years, decades earlier. And unless you know what the history is about the installation and management, you don't know what might be going on. You should know your weather conditions for the last 1 or 2 years. I mean, I think this is becoming more and more obvious as we go on, as we have all these hugely different rain levels or not rain levels or temperature extremes or whatever. We had flooding in the Northwest this year, which was a 100-year flood, which is becoming more like a 20-year flood. But it's something that was really different, and it will show up in tree damage in following years because some trees will tolerate it, other trees don't. Look at the details of the damage you see, if there are easily seen details. Is it something that's acute, all of a sudden it shows up? Or is it something that is just there year after year after year, and that would be chronic? And being able to diagnose between those two things is important. And finally, is it caused by something biotic living, or is it caused by something environmental, a non-living cause? So these are things you just kind of get familiar with. You can't do this from a picture. You can't do it from a sample someone brings you. You have to be able to see the site. Then we need to think about hereditary hijinks, for lack of a better term. And this is because we have a love for unusual looking plants. And so we love foliage that looks different than normal. And lots of people like to have trees that are almost pure white with just a little bit of green. I think, because I'm a physiologist, it always looks sick to me. But it's something that a lot of people like. It's aesthetically pleasing. So if you have a sport pop-up, like here, where you've got a green leaf that's popped up, it's important to understand and to explain to your client that this is the wild type of this plant. It's reverting to wild type because the green leaves are much more productive and obviously are going to compete more for resources than the ones that are white, which are not very photosynthetically active. So you have to know you have to take those things off, or eventually those types of sports are going to overtake the entire tree. You've seen this probably before in a lot of dwarf Alberta spruces, when you get those wild hairs coming up. It's just the tree going back to its original nature, and just knowing that helps you manage it, and it helps you explain to your client why it's going on. Then we've got natural variations in bark, which we love, again, because it's aesthetically pleasing. But lots of clients will think it's damage. They'll think that it's bugs or disease. And so knowing the tree, knowing the species, knowing what it normally looks like, you can explain, no, this is just something that looks really cool, but it's perfectly normal for this species. And finally, the really cool mutations like camperdown elms, and how many people know the story of camperdown elms? Oh, not very many, probably because you don't grow them here. Anyway, the camperdown elm, that weeping form, was discovered in Camperdown, England. It was isolated by a single branch that was creeping along the ground. It was cut off and propagated vegetatively, and every single Camperdown elm is a graft onto a normal elm tree because it does not propagate sexually. So every time you see one of these, it came from that original branch that was found in England. So it's really cool. Then weather issues. My garden is a good test spot for this, at least the garden I used to have in Seattle, because we were at the bottom of a hill and cold air would always come and sit down at the bottom of the hill. So we were much colder in our little pocket than actually the temperatures farther up the hill. So when gardeners or property owners see this kind of damage, this is a rhododendron, and you can see that the flower buds have turned brown, it's because we had a late freeze. So this happened in April, at which time all the flower buds, and on some plants, the leaf buds were expanding and starting to grow. Now these flower buds, 2 months earlier, could survive minus 40 degrees. But now, they got a little freeze of 30 degrees and they died. And the reason they die is because when a bud is expanding, it's filling with water, the tissues are not protected because you can't lay down your protective cuticle until it's finished expanding. So it's really sensitive to temperature differences, water differences, and when you get cold freezes, it just freezes like an ice cube. And so they all die, but the leaves you see are fine because those are last year's leaves and they're protected. So when you see this, just explain what's happened, that the expanding tissues are really sensitive to both heat and freezing, and then it happened next year, hopefully it won't happen again. But those tissues are toast. But they're not a disease, they don't have to be sprayed, it's just nature. Okay, and so here's an example, same plant, different year, where the leaf buds were expanding, and this also was in April or May, and we got a heat wave. So for us, a heat wave at that time of year is anything about above 60 degrees. It was 80. So what happens is these tissues, again, they're not adapted to conserving water. Their water evaporation was much faster than the water uptake, and the leaves got all that browning on the edges and margins and tips where water was being lost because there's no cuticle to protect them. So then they turn brown, but the problem is if you show this kind of picture to a homeowner, or it happens to them, the first thing to do is to get online, right? Look for pictures, look for AI to tell them what's happening, and they'll be told probably it's some kind of nutrient deficiency that's caused the warping and cupping and browning. It's not. It's weather. And it's really important Well, AI just sucks for what we do anyway, but pardon my language. But you can't diagnose these problems just from pictures. You have to know the circumstances. So understanding that the leaves are not protected when they're expanding. Once they're fully expanded, they lay down a cuticle, and then they are much more tolerant. Because you can see the underlying leaves from last year are fine. And then if you have excessive heat or drought, believe it or not, we do have excessive heat and drought in the Northwest, and always have. We've got a Mediterranean climate, but now it's hotter and drier than normal. And you have this chronic dieback of the leaves, starting at the tips and margins and working back. That's telling you that for some reason there's not enough water, either not enough irrigation, too much competition, poor root system. There's a lot of reasons it can happen, but that's what's causing it. It's just a lack of water in the tissues, not necessarily lack of water in the soil. It depends. Then there's all kinds of horrendous things you'll find at nurseries and other places, like this lovely plant from Seattle's most expensive nursery, where it had a really nice root system before it was hacked to death and then stuck in a plastic bag that contains virtually nothing except some fine roots. But the structural roots are gone, and it's so hard to find nice structural roots now anyway that seeing this was just doubly painful. Putting things in the wrong place. And so this tree, it's hard to say exactly why it's suckering, but the suckering is an indication of something going wrong at the roots. All right, so if it's a species that doesn't normally sucker, and it starts to sucker, then something's wrong, either not enough water, poor root system, too much evaporation, and not enough uptake. It's hard to say exactly. But when you have a tree sited in a hot area, and it's got a stone mulch with heat being reflected off of it, with heat being reflected off that stone building, then you're going to have a stressed plant, unless it happens to be resistant to that. Young trees in general aren't resistant. They lose a lot of water through their trunks. The bark tends to be pretty thin. It's not protected. And so you can see both that the bark, well, you can't see very well, but the bark is damaged, and you've got those suckers coming up. So the tree's not happy. And sometimes with enough chronic damage, you will get a tree that looks like this. And again, homeowners will go online, they'll type in, what's causing my tree to die back? And they often get verticillium wilt. This is not verticillium wilt. This is chronic heat damage year after year after year. And what happens is the tree's putting on new growth in the spring, but what happens in the summer when it gets hot and dry, that new growth dies back. And that happens over and over and over again. And so the tree's basically shrinking. And it's losing more and more of its top growth. But it's not a disease, it's because the tree is not in optimal conditions. We don't know why, because we can't see what's going on underground. So, with that as just kind of some of the baseline things to think about before you do a diagnosis, here's case study number 1, allelopathy. And I'm going to assume everybody knows what allelopathy is, right? Maybe? And so this is something you will see online, and if you type in allelopathy in black walnuts or juglone or whatever, you will find thousands of pages on this. And so what the message is that juglone that's in black walnuts will kill underlying vegetation, whether it's grass, other plants, whatever. So you shouldn't have it in a landscape, especially a landscape where you don't have enough water, because it's allelopathic. And thousands of plants since this have been found to be allelopathic through a lab test that I'm going to show you. So there's thousands and thousands and thousands, but juglone, black walnuts, has been tagged as the biggest problem of the lot. So let's look at this. This is how you test for allelopathy. So you can do this with any plant extract, and you have petri dishes. So A is a petri dish with just water in it, and then B through G is increasing levels of some chemical, in this case juglone. And once you get to where these germinating lettuce seeds start turning brown, there at the bottom with E, that's when the tissues are dying. So because this is all done in very controlled conditions, and you've got these increases in concentration, you can say with certainty that the concentration in E is the lethal border, or where lethality will start. So anything above that is going to die. And you can do this over and over and over and over. It's repeatable, it's scientific, it's good information. And as I said, you can do this with lots and lots of different plants. So many plants make allelopathic chemicals. But the first one, again, was in the early part of last century when scientists were looking for ways of having natural herbicides. This is when people were starting to worry about the chemicals we were using and their toxic effects outside of killing whatever pest you're after. And they wanted to be able to find natural chemicals to kill plants, to kill insects, whatever. So juglone was one of these things, was isolated, identified, and found to be allelopathic. In other words, kill other plants. So perfect for an herbicide, right? So the scientists are studying this back in the 1920s and studying it consistently for several decades. And it was tagged as being the problem initially with killing tomatoes. And so the story is, I call it a story rather than the facts because I've not read the original transcript, but what I've read is that one of the scientists studying this has his eyes opened to what allelopathy is, goes home and sees the tomato plants near his walnut tree, as in that second picture, the ones closest to the tree were dying. And as you got farther away, they looked healthier. So he called this tomato wilt, some people called it walnut wilt, and this was used as kind of the metric for demonstrating that black walnuts will kill other plants they're planted near them. So scientists are really excited at this point because they're finding these chemicals that will do naturally what we were using synthetic chemicals to do, or toxic heavy metals and things like that. So this was really exciting. The black walnut and other walnut growers in the meantime are like, hey, we grow these things and there's grass and other things that grow underneath them. You're making people afraid of walnut trees. Because as it turns out, all members of the walnut genus make this chemical. It's just black walnuts, for some reason, got tagged as being the bad guy. So juglone is contained in the roots and the hulls around the nuts. They're not in the leaves. They don't get spread in the air. They're only in two places, the roots and the husks of the nuts. And in the 1950s, as I said, as they're working on this, they confirmed that juglone, they confirmed its chemical structure after they isolated it, confirmed over and over and over that yes, it will kill germinating lettuce seedlings in Petri dishes. And then, based on that, they said, these are the scientists saying this, that this is what kills plants in the field. Okay, so going back to the picture of the tomato plants and the walnut tree, so no field research to show this, but the fact that it works in the lab was then way over extrapolated to what happens in nature. So this is where the scientists started the whole conflating correlation to causation. But the problem is, as many of you might know, especially if you spend any time in Canada, where black walnut trees are a really common landscape tree because they're quite cold hardy, they have gorgeous gardens underneath them, right? So how do you explain this? And okay, so I'm a plant physiologist, and I should have been asking this question of myself 30 years ago. I did not. But how do you explain it? What's your hypothesis for why this happens to some plants, why it kills some plants but doesn't kill other plants? And there was no really good mechanism for this. So what scientists did instead is they started creating these plant lists. And you can go online and you can find the plant lists. Big, long list that tells you what plants are sensitive to juglone, and they include everything from herbaceous perennials, trees, shrubs, annuals, and vegetables, to those that are tolerant to black walnuts, which includes trees, vines, shrubs, annuals, vegetables, fruit trees, and herbaceous perennials. There are no differences between these groups, right? I mean, you should be able to figure out a biochemical pathway that exists in some group of plants that allows them to become tolerant when other ones don't have that. And that happens all the time with other types of stresses, but it doesn't happen here. So it makes it really hard to defend, but the lists are there, so we all rely on the lists. So I started looking at this a little bit more objectively about 8 years ago to try to find where did the data on the lists come from. And I went back through all of these journal articles, and they all repeat these lists, which were getting longer and longer the longer they were around. But they all pointed back to an original paper in 1973 that was called Walnut Wilt, and that was out of Cornell. But I couldn't find data for any of these observations. So these lists were apparently created solely by anecdote. You know, oh, I saw this happening, and then it was put into a list, which scientists aren't supposed to do, right? But these lists are repeated and published in peer-reviewed scientific journal articles because authors did not do their due diligence and find out where the heck did this come from. They just repeated it because it was someplace else. That's a real no-no if you're a scientist. So I kept on going back and back and back, and I finally found, you know, the reference to this 1973 paper. It was called Walnut Wilt. And I couldn't find it online, so I contacted a graduate student of mine who now is a professor at Cornell, and I said, I need to talk to someone in Extension that might be able to help me find this paper because I can't find it online. So she put me in touch with the right people. They spent a couple weeks, got back to me, and she said, we don't have a publication called Walnut Wilt that was ever published in 1973 or any other time. We don't have anyone on faculty whose last name was Christ. We don't know where this came from. So all of a sudden we have nothing. However, this story, and this is not in the publication that I made from this, my fact sheet, because it's too new, but in the last few months, I got an email from someone who said, I found the paper you're looking for. And here it is. So this is a single page, not from Cornell Publications, but from Schenectady County in New York. So it was, and this is the way it's been cited in peer-reviewed journals. There's the citation right there, and you can find it all over the place. It says it's a horticulture extension bulletin from Cornell University. It is not. It's a master gardener project from Schenectady County. And you can see this, I've got it highlighted down there. It says master gardener / the whole thing for the URL, projects. The author is a extension aide, which is, I think, what they used to call master gardeners. So this was just a one-page blurb, that they put up on, that they had online eventually. Initially, they weren't because there was no online in 1973, but it's gotten up there now as part of the county's files. Non-experiment has no data and no references. Yet everyone cites this as being the publication that is at the root of the whole allelopathy story. So this is really kind of shocking to me as a university professor who used to teach about allelopathy in her plant physiology courses, that it's all built on nothing. Now, it's not to say that you can't do it in the lab, because you can, but it does not happen in nature. And that's why for you all who are practical people and working on landscapes, don't worry about allelopathy. It's not a problem. So, all the plant lists are based on observation, not experimentation. There are no physiological reasons why some of those groups would be sensitive to juglone and other ones wouldn't be. And the negative effects you see, if you see them, are probably due to something as boring and uninteresting as you don't have enough water or you don't have enough light. So that's the most likely reason, because people have tried to isolate juglone from the soil and they can't find it in concentrations that would cause damage. So it doesn't happen in nature because juglone is an organic compound. It gets broken down really fast by microbes. So it doesn't happen in a petri dish, no microbes. It doesn't happen in controlled greenhouse experiments where you're using sterile soil. But in nature, where you have microbes to take care of things, it doesn't happen. So other people have made similar comments, maybe not as pointed as mine. Like, while the genus Juglans provides the most widely accepted example of allelopathic plants, there is still no unambiguous demonstration of its effect. And as juglone is rapidly metabolized by soil bacteria, it is unlikely to cause any appreciable effect. And juglone is not particularly mobile in soil. It is readily absorbed by organic material. It doesn't have an effect in nature. So right from starting today, never, ever, ever tell a client that plants are being killed by chemicals another plant is making. because it doesn't happen. And even now, I mean, I still get questions about eucalyptus and other things, and I go back to literature. There are no field experiments to show it happens. You can do it in a lab with a petri dish, with germinating lettuce seedlings. It doesn't happen in nature. So how do you avoid, quote, allelopathy? Make sure you have enough water for everything that's planted there. If you have dense plantings, you're gonna have more competition. You're gonna have more death. If you don't use irrigation, you will have more plantings. New plantings die with the tree surviving because it's bigger and better established. Wood chip mulches, if you have trees, you gotta have wood chip mulches. Wood chip mulches will retain moisture and help those plants live. And I know this isn't a big deal, obviously, so much in where you're from if we're talking about the desert Southwest, but there are places like your gardens and things and public gardens and arboreta where you do have collections. But the black walnut is not going to be a problem there. Or you reduce light competition by planting full sun plants other places and putting the ones that do tolerate shade underneath the tree. So that's just, it's normal plant selection. It doesn't have anything to do with juglone. So anyway, if you want to read more about it, I've got a fact sheet on it. But this is just the beginning of talking about not conflating correlation with causation. So there's a wonderful, wonderful website called Spurious Correlations, and it just gets better and better. But you can see what this is correlating almost to exactness, eating cheese with dying in bed, right? So if you don't want to die by becoming entangled in your bed sheets, don't eat cheese, which is basically what this chart would say if you were saying something was causing something else. So he has tens of thousands of these, and this is a good way. The website is a great way to try to get someone to think outside of where their head is and look at, sometimes things are just correlated. One isn't causing the other, and you have to show one's causing the other. So here's the fact sheets that you might be interested in. At the end, and also on the handout, there is a link to where I keep all my fact sheets online. They're all peer-reviewed. They're all free downloads. One is on the black walnut tree and allelopathy. One's on arborist wood chips, and then on mycorrhizae. And so all these things are connected together. Okay, case study number 2, nutrient deficiencies. So again, really important to have soil tests. And we'll talk about fertilizers in my second talk, which is right after this one. So I'm not going to go into a lot of detail about this, but I will in the next talk. So you see things like this where you've got yellowing leaves, either the older leaves are turning yellow, or perhaps you've got intervenal chlorosis where the veins are green, but the tissues are yellow. And you use that as your guide for adding fertilizer. And I mean, I will tell you that so many of the things that I talk about are things that I did and believed, even with my PhD in horticulture, because I was doing lab-related things, a lot of theoretical research. It was not practical on-the-ground research. So changing what I did as a professional really changed the way I looked at many of these fallacies. So if you were considered this to be a crime scene, your first thing to do would be to look at a lab analysis, right? So here's a soil test. And if we look at this, we can see that, come on, there we go, that the iron is 4.2, and the range is 2.7 to 9.4 parts per million. So the iron in the soil is within normal limits. But the iron in the leaf is obviously deficient, or manganese, depending on which one it's from. And you don't know until you do the analysis. So it's not from a lack of soil iron or soil manganese often. What you do instead is you look at your phosphorus levels. And you can see phosphorus in this case is at 45.2 parts per million, and it's supposed to be between 4 and 14. Now that may seem low to you, but you have to realize that a soil test for a landscape is not the same as a soil test for an annual crop field. Because landscapes recycle about 90% of their nutrients. So we don't have to have very much in there. So 4 to 14 is ideal, and this thing is 3 or 4 times higher than that. And it turns out that phosphate actually interferes with both iron and manganese uptake at the roots. More phosphate you have, the harder it is for the plant to get iron and manganese into its tissues. So before you say iron deficiency, we need to add iron chelate, find out if it's phosphate. Because you can add all the iron chelate you want, and if you've got too much phosphate, it won't make any difference. So that's why one reason why the soil tests are so important, because you can diagnose these problems, and you've got data to back up your diagnosis. So the problem is that you've got excess soil nutrients that will cause leaf deficiencies from fertilizers that are too high in phosphate. And the problem is that all of us, homeowners, professionals, everyone has been sold this, which is a agricultural recommendation that when you plant, NPK, nitrogen, phosphate, potassium, those are your deficient elements and you have to add them all the time. Well, we're not doing crop fields, and the only thing that's ever deficient on a regular basis is nitrogen. Phosphate is hardly ever deficient in managed landscapes. But you will get this problem with the chlorosis, as the phosphate is going to reduce the uptake of iron and manganese. So you let your soil test tell you what's going on. If you have a soil test that tells you that everything is above optimum, don't add anything else. And if you have a soil test that says everything's at optimum, don't add anything else. If you have something that says it's very low, that's an indication to add that something. But when you're looking at a managed landscape, you're hardly going to find anything that's deficient except nitrogen. And most of the time, we don't even measure that because it's so variable. But you'll see lots and lots of things that are off the scale the wrong way. So, commonly excessive nutrients include phosphate, magnesium, and calcium. Low does not equal deficient, and we'll talk more about those three nutrient problems in my second talk. But if you have something that says low, and in this case the low is 3.4, and the range is 4 to 14, it doesn't mean it's deficient. It's when you have to ground truth your results. So I'll tell you right off the bat, this is a soil test from my landscape. Okay? Everything is optimum except phosphate's a little bit low. So what you do is you go outside and you look. And it turns out the trees are fine, the landscape's fine. The only place I ever found a deficient level of phosphate was in my vegetable garden, which is like crop production, right? Because you're taking the whole thing out. And I started to get tomatoes that had symptoms of phosphate deficiency. So that's when you add something. When you ground truth it and you see deficiencies. If you don't see deficiencies, don't add anything. So figure out the evidence of deficiencies, whether symptoms, whether nutrient toxicities. You can oftentimes connect these together. Phosphate toxicity is the one that we have the most evidence for so far. It's difficult to do this kind of experimentation, and so the robust literature on other toxicities, things like magnesium and calcium, we kind of guess what the problems might be, but we don't know for sure yet. So here, again, some peer-reviewed information, one on phosphate, one on Epsom salt, one on gypsum, and those are free downloads. And again, I'll talk about these a little bit more in the next talk. So case study number 3, volcano mulching. Who has not heard of volcano mulching before? I knew it. Oh, one person. Okay, so you've all seen various types of volcano mulching. And I will say, first of all, I will never recommend that you volcano mulch, ever. And I can't even tell you what's going on with that. Well, I can guess what's going on with the third picture. So that's at a flower and garden show, and I think it was in Sacramento. And I think what they were doing is they were hiding the presence of a box, which that palm tree was probably in, right? Okay, so keep that in mind. We can use mulch to hide things. Okay, what are we hiding? So before we go into that, again, from my best friend on the internet, Tyler Vigen, creator of Spurious Correlations, he says, instead of starting with a hypothesis and testing it, I toss a bunch of data in a blender to see what correlations shake out. It's a dangerous way to go about analysis because any sufficiently large data set will yield strong correlations completely at random. So here's another fun one to look at, and this will explain to you why AI should never be used. But here's the correlation between the votes for libertarian senators in California correlated with the number of moderate earthquakes worldwide. Now, everyone's going to laugh at that, right? Not AI. AI has an answer for you. And AI says, the decrease in libertarian votes for senators in California led to a decrease in seismic political activity. This, in turn, created a more moderate political climate, reducing the frequency of political earthquakes. As a result, there was a ripple effect on the Earth's crust, leading to a decrease in the number of moderate earthquakes worldwide. It seems that even tectonic plates prefer a more moderate approach to governance. So if that doesn't cure you of AI, nothing will. My job, even though I'm retired, is secure. Because so much of what is garbage out there gets picked up by AI and incorporated to whatever answer you're trying to find. So anyway, back to the volcanoes. Are you sure that the volcano is just mulch? First question. Maybe it's not. Maybe it's hiding really bad planting. Maybe there's not even a hole dug. Maybe somebody builds a hole by putting soil on the ground and then making a pit for a ball to go into. This happens all the time, really bad planting, and then it's all covered up with mulch. So as I said, mulch can hide a variety of sins. So other mulches, and as the mulch starts to peel away, you can see bald trees that still have the burlap, they still have the twine. They're not establishing in the landscape, the roots are just sitting there. But if they're covered up with mulch, you can't see that. But the trees suffer, the trees start to die, and mulch gets the blame. Now, it isn't to say that some mulches won't do this, because some mulches might. But wood chips get the blame almost all the time. We also have bad roots. If the bad roots are covered up, you can't see them. Eventually, they might emerge, or the plant might just die within a week after being planted because it had no roots. And all of this is blamed on mulch if it happens to be all covered up with mulch. So without going into too much detail, because it's a completely different talk, we know that we're going to have better plant establishment if we get all that stuff off the roots and get the roots in touch with the native soil like we used to 100 years ago, before containers existed, before B&B existed, and you planted everything bare root. So all the trees you see that are hundreds of years old in developed areas were planted bare root. So going back to that, and this again is not topic of this talk, but just keep in mind that you're taking off those barriers, you're correcting roots if they have flaws, and you're plant at grade. And that will cure many of the problems that we see with trees dying early on. So keeping in mind that a hand in the glove approach will stabilize roots, a ball and socket does not. And so we want to do the hand in glove, we want to get the roots in touch with the native soil. Trees are much more likely to survive in those circumstances. There's research to show, it's been going on for decades, that bare roots ultimately do better than either containerized or ball and burlapped plants. But now let's talk about the mulch. So wood chips being blamed, but sometimes other mulches are used. People will use fine textured mulches like compost or sawdust. If you have really deep layers of that, that will interfere with oxygen getting to the root ball. So yes, if you have deep layers of one of those things piled up against a tree, that could absolutely be killing the tree. The problem is you can't use thin layers of this material because it doesn't block the light, the weeds come back, and so just thin, fine textured mulches are just not a good choice because they don't work and they can cause damage. Things like bark are hydrophobic. They shed water, they don't absorb water like wood does. They have waxes. They are part of the outer part of the tree. Their function is to keep water inside the tree, so their function is not to absorb water and release water. They have a completely different function than wood does. So this is water repellent mulch, and as it gets dry, it'll blow away because it just doesn't ever get integrated with the rest of the soil system. So there's been some work, exactly one published paper, looking at coarse wood chips and ultimately what volcano mulches would do. So specifically, this is looking at what happens if you use a coarse wood chip mulch and then you have a wounded tree. So you can see here that there's a tree that's been wounded. So in this particular experiment, which was done in Minnesota, 40 trees were used, 20 of them were mulched in the regular way, and 20 of them had a volcano mulch applied. And they all had this wound because this is the easiest way to see if this is what kills plants, right? Is that if the pathogens are in the mulch, they're going to get in the tree and they're going to kill the tree. So this is looking at that claim without us having any idea what was going to happen. We're hoping that what happens is that it doesn't, it doesn't make things worse. So what happened, which was completely unexpected, is not only did it not kill any trees, it actually caused sealing to occur faster than if it had been left uncovered. And that was unexpected. So you can see, hopefully in that picture, it's kind of tough to see, but there's where the wood is darker down there, which is an indication of been covered up with mulch, there's a very thin slit, and that's what's left of the wound from the first picture. In the ones that have been left uncovered but mulched, the wound is much larger, right? So they sealed over faster if they were covered with the mulch. Now, we weren't looking at why, but we could guess that perhaps it was because it was a environment that was shaded. It wasn't exposed to sun or heat. It was moister, so that the natural sealing process might go faster. Nothing got diseased, nothing died. There was one tree that died, but it happened to be in the one that was a control that didn't have the volcano mulch. So, unfortunately, no one's repeated this, and it's kind of too bad because it would be nice to show it over and over, but it really is pretty stunning to see that not only does volcano mulch not kill trees, but if you have a wounded tree, say from lawnmower or some other damage, it might help them seal faster. Now, that would be a really cool thing to investigate, to see if that actually would work. So arborist wood chips, we already know from a lot of work, are really good. Deep layers are really good at suppressing weeds, and they're really good at helping trees get established. And so deep layers of wood chips on their own are not going to cause trees to die. It's been demonstrated with research from all over the country, if not all over the world. And if you have fine textured mulches, it will cause a deficit in oxygen getting to the roots, and that could lead to trees being compromised, possibly dying. So fine textured is what people like the looks of. It's not really that great. The coarse textured stuff looks messy. Lots of people don't like the way it looks, but it's very effective in helping trees get established and surviving. Okay, so anyway, if you're interested in, now this doesn't look at fine textured mulches, but it looks at different landscape mulches that get put down as weed control types of things. So this was published in 2019 in a journal that is specifically on soil research, much of it by soil engineers, much of it mathematical. This whole paper is nothing but calculus, except for the end. And so I was the plant person, everybody else was a soil scientist. But what we found was evidence that among all the mulches tested, which I'll show you in a second, that wood chips are a preferred method of mulching in terms of providing best gas permeability, particularly in landscape conditions. So this is compared to all these sheet mulches that we compared it to. So we weren't doing rock or anything because there's only so much you can do, but sheet mulches get used a lot supposedly for weed control, and that's what we wanted to investigate. So these are the data that we came up with. First of all, I want to explain that the axis on the y-axis on the left-hand side is an exponential axis. So each one of those tick marks is a factor of 10. So it's like earthquakes or pH scale or something like that. And along the bottom on the x-axis, we've got bare soil, wood chips, cardboard, landscape fabric, and polyethylene mulch or plastic mulch. And the wood chips were 4 inches of wood chips. The cardboard was a single sheet of corrugated cardboard. Then we had a single sheet of fabric, and we had a single sheet of plastic mulch. And through a really interesting experiment, but just tedious in terms of the mathematics involved. I did not do well in calculus. I will admit that readily. It was very difficult to read the paper, but I was able to extract the data and create this graph, which I think should have been in the paper, but not the soil scientist. But anyway, here's the graph. And it shows you that the bare soil as the control was the best. Now, this is not anything you'd find in nature, because the bare soil in our experiment was in the lab. It was nice and fluffy and well oxygenated. It didn't have compaction forces or anything. It's not real, but it's the standard for the test. The wood chips were about 10 times, if you want to say, worse than having bare soil. Again, knowing this bare soil does not exist except in the lab. Cardboard was 10 times worse than the wood chips. The fabric was roughly 100 times worse than the cardboard, and the plastic was 10 times worse than the fabric. And so this is in the ability of carbon dioxide to get out and oxygen to get into the soil. And so we don't see what's happening underground, but we know now what does happen in terms of these mulches. So if you want to work backwards, you can say that polyethylene is 10, 100, 1,000, 10,000 times worse than 4 inches of wood chips in terms of gas exchange. So with the cardboard craze and everything else, you know, there was no research prior to this paper, and I don't know if there's been any since. I don't think there has been, not since I last looked. But this really gives you good mathematical evidence that sheet mulches just aren't appropriate for trees, because tree roots use a lot of oxygen, and they won't do well if they don't have a good source of oxygen coming through the soil. So anyway, I want to suggest for your clients or for yourself that when you're trying to get someone to think differently about what they're reading, especially AI, although we don't talk about AI in this paper because AI was not a thing right then. It's a manual that will allow you to evaluate information from various sources by looking for certain key attributes as you're reading through it. It's a nice way to start becoming more objective in the way that you're looking at evidence. So if you've got somebody whose opinion you're trying to change, this is really fun. It's got all kinds of interactive stuff on the web. It's written at a 10th grade level. You don't have to be a scientist to read it or understand it, and it's a great way to increase your ability to judge information. So I am cutting this one a little bit short, not too much, but because I do want to leave time for questions. And I do want to have especially time for questions after the second one. So I'm not really cutting it short, but I'm just deliberately let some time for questions because I know that this is a topic that is of great interest. Yes? Okay, so the question is, when we're talking about arborist wood chips, are these aged wood chips or fresh wood chips? Now, there's absolutely nothing wrong with using aged ones, but my colleague in California, Dr. Jim Downer, who's a microbiologist and pathologist, has done research on wood chips as they age, and there's no danger of disease or anything, but what happens during that first month is you get this big burst of nutrients coming through because arborist chips have leaves and needles in them. And so those things get broken down quickly. So having those on the soil as fast as you can means you're getting those nutrients into the soil. They're good at any time, they really are. But he always says fresh is best. So because he's the guy who did the research, I will say fresh is best. Other questions?
Here's the mic.
Yeah, it just came to mind. Oh. On that notion, you said, are there diseases that could be transferred by fresh wood chips if you are, top dressing soil or landscape with them?
Okay, good question. So this is a question lots of you might get from clients about using fresh wood chips and disease transfer. So let's say you have Armillaria, Phytophthora, or something else, some kind of rot, root rot, butt rot, whatever, that's in that wood. Will that cause a problem? And the nice thing is that there's been research looking at that, and the answer is almost uniformly not a problem. Because you have to have three things, obviously, for the pathogens to be a problem. You have to have the pathogen, which you've got as a spore. You have to have the host, but then you have to have the right environmental conditions. And because wood chips and the soil under them is so oxygenated, the pathogens don't grow under those conditions. So the beneficials will grow under high oxygen conditions, but not the pathogens. Now, I will say that if you take the wood chips that are infected and mix them into the soil and damage roots along the way, you can absolutely transmit diseases that way. So the wood chips should never be used as a worked-in amendment. They should only be a top dressing, as a mulch. But no, the disease issue has not been shown to actually happen with diseased wood chips.
Question over here.
Hi, I've been using phosphorus as a plant healthcare tool. There's a lot of phosphorus products out there. What is happening? Do you think it's working?
It's a really good question because I know I think it's mostly phosphites that are being used for disease control and stuff. I have not seen anything to suggest whether that makes the overload of phosphate as, just the element itself worse. But I really would be curious to know. And the problem is that most time when that happens, there isn't a soil test being done at the same time, so you don't know what the phosphate level is. I would be possibly concerned. It would be really nice for the people that manufacture it to sponsor research to demonstrate that it doesn't make the phosphate level higher. But it's a question of concern.
Question here.
On the same point as a fungus, but isn't it going to draw insects to the bark?
Oh, to the wood.
To the wood, yes.
Yeah. Bark is. So, and that's a good question too. So, are wood chips something that are going to attract insects? And I think people worry about termites probably more than anything else. As far as most termites, and I know there's exceptions to termites, but most termites will not eat wood chips. And in fact, in choice experiments where they've actually tested what will termites eat, wood chips are at the bottom because there's just a lot of stuff in there that's not palatable. I mean, not only do trees manufacture allelopathic things for plants, they can manufacture allelopathic things for insects. So what termites really like, what the control is for these is cardboard. That's their favorite food. So that's another good reason not to use the cardboard. But so in answer to your question, there's been no evidence to show that that insects will eat it. Now, whether it houses them or not, I suppose, is a point of argument unless every single insect has been tested. And I think I saw something that possibly fire ants would get into wood chips, but I don't know because fire ants are not something that I deal with. You guys know about stuff like that. Anybody seen wood chips have fire ants in them? Okay, so maybe not. Maybe not.
Was there a question over here? Questions. We got about 4.5. Oh, we got a question.
Earlier in the presentation, you talked about not adding phosphorus supplementation because 90% of the nutrients are recycled within a managed landscape. Is that hold true in landscapes where they're frequently removing leaves and frequently removing needles so that the organic material doesn't have time to break down?
That's a good question. And in answer to it, yes, that would decrease the percentage that's recycled because it's being removed. But phosphate has never been one of the things that's been found to be lacking in most urban sites tested, and there's been soil scientists all over the world now that have looked at this and said that the number one problem with urban soils is phosphate overload. So just as a comparison, we did a similar experiment in Puyallup where we tested 200 residential sites, many of them from master gardeners who volunteered to do this, just to look at phosphate levels because there's a very important watershed that's in Puyallup. And so 200 sites, the lowest amount of phosphate tested was, I think, 38. And again, the range is 4 to 14. And the highest was over 900. So it's just a huge problem. And it's one that most homeowners don't realize they have because they're used to following these guidelines that say, when you transplant, you must add phosphate. And it's just not true. So, if nothing else, hopefully you'll get intrigued enough to have soil tests done on your own landscape, if nothing else, just to see what you can find out. I mean, I will admit that I didn't do any soil testing myself until we moved out to our family farm, and I was going to grow vegetables, and I wanted to know what we had. And so I was pleasantly surprised to see that ours, because my parents really didn't ever add anything to the soil, it was optimal, except for a little bit low in phosphate. But it's really instructive to do these and then see how it plays out in your landscape.
I think we have time for one more question. Was there over here? Okay.
often cited in the reasons to not volcano mulch are reports of the trees wanting to create adventitious roots that could cause problems later. What are your thoughts on that?
I think that if you've got a tree that's planted too deeply, number one, and you can't tell, especially if it's volcano mulched, or it has a fine textured mulch, then absolutely it's going to try to put out adventitious roots. But a tree that's planted at grade, that's mulched with wood chips, which is very highly oxygenated, is not going to do that because it doesn't need to. Well, good questions. Thanks very much, and I know we've got one more slide for you. Oh, there it is. There's the code. I'm the next speaker, so we'll have a minute for me to change my slide, the slide presentation over. But thank you very much, and we'll continue on with fertilizers.